Right. So we stopped here. We looked at still on the Gustavo Maroni textbook. So here we looked at an example of dominant disease where I showed you that, well, you can also show that it is possible, that it is you can draw the alleles and say that it is also recessive. It's possible, but it's not probable. You should know the difference between possible and probable. It's possible, you can do it, but it's not probable because diseases are rare, and likelihood of spouses from the population to be a carrier is not very likely. For example, if you have cystic fibrosis, for example, you're a carrier. When you get married to other people, it's unlikely. The chances of suddenly you marrying someone else who also is a carrier on cystic fibrosis is normally very, very low. All right, it does happen though, it does, but the chances are very, very low. So normally in statistics we will calculate that. We will normally assume, if you don't have any other information, you'll assume that the spouse is normally normal, okay, or unaffected, sorry. So okay, we continue with this book. So now it gives you an example of autosomal dominant disease. Again, you should be able to. Now, this is one example. If you read this, then it says C1 is a marker locus. Marker means that is not the disease locus. It is very close to the disease locus. It's a marker, because sometimes diseases are not variable, so you cannot do genotyping, but we'll talk about the variability of markers later. But we use C1 as a marker, as in if you have C1, that means you will have the disease gene. So C1 is a marker locus. You'll find that, okay, this one has got the C1, and then C1 here. Whoever is affected has got the C1 marker, so that sort of. But you go down further, it says here's a C1. This is a consanguineous marriage. Remember what is a consanguineous marriage? Marriage between relatives. So you have a C1, and then you have but they're not affected. What's going on? Read further. You'll say that it says somewhere here. Let me change that into a highlight. Okay, it says HD is a neurological disorder caused by dominant mutation, but carriers of the gene remain asymptomatic until middle age. What does it mean? It means that you don't see the symptoms until you are late. You have grown further in life, in your adulthood, that you can start to see the symptom. So chances are this individuals right here are young, so you don't see them yet. So but for the purpose of counseling, you probably can tell them, you have this, you can meet up with the family and say, oh, because you have done a consanguineous marriage, you're married between relatives, the chances of the alleles of coming together is even higher. So these are the chances that you may get. You can calculate the risks and so on. So you can tell them that now your son or daughter, because again they don't know, maybe they didn't, the information was not available. Say that, oh, they have actually inherited the marker locus, so it means that they probably will get Huntington's when they grow older. So you can say that. All right. Now, examples of autosomal recessive, and so on. Okay. Now, I'm going to skip all that. Again, it's talking about familial studies. What I want to do is, now, this is, why do we study pedigrees? You want to calculate probabilities. And for this, I will go on, later on, I will go into another note, the human pedigrees, but before I go, we'll just finish up a few more parts in this book. So probability of individual genotypes, you've done this before. I think at some point you calculate what is the probability of this person inheriting. So here you can actually do a little bit of calculation. Let's just enlarge this a bit. Now this is what? This is autosomal recessive, sorry, X-linked recessive trait. So you'll see that this individual has got the small c, it's an X-linked, so he's got it. And no male to male, right? If it's an X-linked, you don't see male to male transmission. But now the daughters are carriers. Again, so this is somebody from outside. You don't expect them to see to get that, so chances are you don't know whether, because if it is a son, then what is the chance? That son has got a 50% chance of getting the small c. You can calculate that. What about this person? Now for that person is affected, so you know he has inherited the small c, and then the wife from the father, the small c. But what about this person? You don't know. She could have gotten the big C, definitely, because she's a female. She definitely got a big C. So you don't know whether she has inherited the big C or small c. So she could be a carrier. And because you don't know that, you don't know whether the son is going to get it, you can calculate that as well. How you do that? So okay, you can calculate that the probability of the mother being a carrier is 50%, or the is 0.5, right? And the probability that out of her being a carrier and passing it to the unborn child is another half, so that means it's half and half, right? So now chances of the son being affected is 1/4. So you can do that, calculation. It's not really difficult. It's just probabilities. So that's why we do pedigree analysis. Okay. Now, then we go into probably group outcomes. This goes to your statistics class, I think, where if two events, the probability of both events occurring is this one times this one, but if you have a probability of two events, A or B, then that is the rule of multiplication rule, and then the one addition rule, that kind of thing. So it's the same thing here. It's the same thing applies, but as long as you know what to do with your how you did the pedigrees. I'll skip through now. There are two things I want to mention here, which is actually the actual main topic of this video, which is ascertainment errors. When I was studying this course, this becomes a big issue. In fact, this is one of the reasons I've actually included this part, because you need to know this issue. Now, let's say you later on you work in a okay. Let's say you work in a genetic study, right? And you're trying to see a dominant land. Let's see a dominant disorder. So you get families coming in, and then you get the data, and then you design an experiment. Okay, let's say I want to know whether this is dominant or recessive. And basically what you do is that the hypothesis is that if you have enough matings of the you can calculate if you have because diseases are rare, right? Remember, genetic diseases are rare. Affected fathers will be D1, D2. If they are married to a normal spouse, will be D2, D2. Okay. So chances of the children to have it is half and half. Can you get that? Right? Okay. We'll try to work it out. If you can't see it, I'll draw it down. So if you pull data from multiple families, if you say let's call all the families who have this trait and count all the children, right? You would expect, because you define that the family has got affected father or mother and an affected child, you will hopefully get, statistically, if you have 50 children, you would expect half of them to be unaffected, half of them to be affected. And this is just by simple probability, right? When you have let's use big A small a crossed with small a normal affected individual, you will get two genotypes, big A small a and small a, and the chances are, if you do that, probability of getting big A small a is half, and probability of getting small a is half, and that for a population you will get that number as well. However, the issue comes when you are trying to do this in a practical manner in a clinic situation, where you try to gather all the families and try to and you start asking each family how many children they have and so on, and then you want to try and get the estimated ratio of how many affected against how many normals, hoping that if your hypothesis is correct, you get 50-50. Unfortunately, the problem that may occur is that in a lot of cases, unaffected individuals or families who do not have any affected children, they don't come to the clinic. All right, they don't even probably know about the disorder, because it has never occurred in the family. It just so happens that the affected father who has probably a mild disease or whatever has not passed the affected allele to the children, and therefore, even though they have 3 siblings, all of them are normal, and so this will not come to the clinic. And because of that, when you count the total number of children, the counting becomes erroneous. That means there will be you don't get the true sampling representative, right? It's not an easy concept to digest. Now look at this, okay? So if the 4 types of families of, if you gather all families with only 2 children, right, in parents, in a situation where one of the parent has the trait, so the parent cross will be big A small a cross with big A small a, correct? If you can get this, all right. So all of this cross, right, you gather all the parents where one of them is affected, one of them is not. Hypothetically, when you do your calculations, you potentially will get 4 types of families. One is that where the first child big A small a, second child big A small a. Are these affected? Yes. So both child are affected, right? You can get the first child big A small a, second child small a. So how many, in this case, how many child is affected? 1. 1 is unaffected. Or you can get the first child unaffected, the second child affected. These are the possibilities. Or you can get both unaffected. So you total them up: affected, non-affected, affected, non-affected, affected, non-affected, affected, non-affected. You'll see that total you'll get 4. This is a theoretical number. So that's why I say, if you gather 50 different families and so on, and then you count all the children, you would expect to get 50-50. However, there is a tendency that you may not have counted this family type 4. There's something wrong with this book. It says, would we sentence. However, if we only counted families with affected children, as this is who would come to the clinic, isn't it? People unaffected will not come to clinic and say, hey, please come and check me, I'm not affected. People don't do that. People who only come for studies are normally those people who come because they are concerned of a particular disorder, or they are affected, so they have it. If you don't know, they don't come. So that means to say, what you have done subconsciously is perhaps collect only families that know of the disease and only have this one. So you have left out families without any history, right? Although the parents may have that history, but because they don't have any children, so their normal children or their unaffected children have not been included in your study. And because they have not been included in the study, of course your statistics will go haywire. Alright? So it says, that means to say you would fail to count 25% of families. What's wrong here is that because it says family type 1. That's not right, because type 1 is both affected. Type 4 actually, I think what it means is that type 4 is unaffected. So this will usually would have not been included in your study. So because that has not been included in the study, then you will actually get a data that shows 4 to the 4 to 2. And you when you calculate, you think that there's a higher chance of getting the portion of affected to unaffected is 2 to 1. All right. Same thing with when you do recessive trait studies, right? Again, the mathematics is here. All right. If you have a family where both parents are carriers, right? Right. These are both parents. B is a normal allele. Small b is affected one. So there are 8 instances. You will see a family that has got all unaffected. You have families with 1 affected child and 2 unaffected, and the order could either be unaffected, unaffected, affected, unaffected, affected, unaffected, affected, unaffected, unaffected. So you can get a family, you're trying to see families with 3 children, so you either get all unaffected, 1 child affected. This one is the probability of getting 2 child affected, so that's one. So you have unaffected, affected, affected, unaffected, affected, affected, unaffected, affected. It's getting confusing, isn't it? But never mind, bear with me. Affected, affected, unaffected. All right. And you also get a family that has got all 3 children affected. And the statistics of that is that you will expect to get in 3 family where 1 affected, you expect to get that in 27 out of 64 families. Families with 2 children affected, you expect to get that in a frequency of 9 over 64, and a family that has got all 3 unaffected, that's 1 over 64. That means if you count this, you get 37 or 64. Now again, what is the issue here? This part. Families in which all 3 children are unaffected. This will not come to the clinic. This will not come to the study. I'm talking about reality here. When you want to do a genetic study and so on, people with do you go to a clinic and ask the doctor whether you have any particular disorder? No, you don't. If you don't have any disorder, you will not go to the clinic. So your data will have not included this group. And because you have not included this group, you've only counted this, which gives you, if you count only this, and then your ratio will be 63 over 48. You don't get 3:1, right? Yeah? So these are the issues. These are called attainment errors, and this is more of a cautionary clause where, you know, it says if you collect data, you make sure that this doesn't happen, because otherwise you will tend to make the wrong conclusions if you want to try to determine the inheritance pattern. All right, so that's the purpose. Again, let me just conclude. You can conclude, and you do some statistics to determine whether it's dominant or recessive. Simple Mendelian statistics, but you have to make sure that your data collection is inclusive, because there is a lot of tendencies that you will not include individuals that do not show the trait. Okay, so that will be for this video. I will continue in the next video.